Hello, everyone. Good afternoon. My name is Aadpaika, and I'm a postdoctoral researcher here at the Yonsei University College of Dentistry. So today I would like to talk about my project on epithelial plasticity enhances regeneration of trace receptor cells following nerve injury. So when I began this project, my professor handed me this small excerpt from a review paper which was published in 2012. So it states that our knowledge adds to the understanding of the balance between the two opposing forces, one promoting the undifferentiated state and the other promoting the commitment to differentiation. So if I put this in simple words, what does it mean? Is that there is a population of cells which we know as stem cells that are maintained as undifferentiated state, and by the form of external signals, they undergo differentiation. You get differentiated cells or the functional cells. So this is what we know so far. But in today's talk, I would like to talk to you about what would happen if this balance is broken by external factors, such as injury. So the model organ which I work on is an epithelial organ. So I will just give a brief introduction about the epithelial tissue and the epithelial organs before I talk to you about my results. So epithelial cells are the polarized cells having the apical side and the basal side. So the apical side has all the functional receptors and the functional structures such as the microvilli or the cilia. And these cells are held together by tight junctions. And hence, because of this, the epithelial cell form a dynamic barrier between the external environment and that of an organism's interior. So because this is the first line of defense for the organism, the epithelial integrity and homeostasis is of utmost importance for the organism's survival. So since epithelial cells form the first line of defense, there is a high chance that it gets damaged regularly. And these damaged or dead cells are replaced continuously through the process called as homeostasis. So like for example, each epithelial tissue have its own definite lifespan. Like example in intestine, it is around for 3 to 5 days. A skin is for like 28 to 40 days, and salivary gland is like 50 to 125 days. So during the renewal process, the adult stem cells are the main factors which drive the renewal and repair of this epithelial tissue throughout the life. So one example I would like to give here is about the small intestine, because most of the vigorous studies are done on this organ. So if you look at this figure, the small intestine can be divided into two parts. The top, the villus, at the bottom is the crypt. So in the crypt part are these LGR5 expressing stem cells, which differentiate into proliferating transient amplifying cells. These transient amplifying cells further give rise to different mature cells having its own different functions, like goblet cells, stuft cells, and enterocyte cells. And these LGR5 stem cells can self-renew and hence can maintain the homeostatic conditions of the apoptosis as well as renewal into the intestine. So in my study, I work on the taste bud and taste receptor cells. So I'll just give a brief introduction about the taste buds. So this is a figure of the human tongue. And on the human tongue, you can see there are these bump-like structures on the anterior tongue and in the posterior tongue. These structures are called as papillae. So these papillae has this spear-shaped structure called as taste bud. So if you look at the magnified image of the taste bud, each taste bud has its own different types of taste receptor cells. These taste receptor cells can mediate the 5 taste modalities, that is salt, sweet, bitter, umami, and sour. So the function of this taste receptor cell is to convert the signal from the taste sense to the electric signal and pass on to the innervating nerve. So the posterior tongue has the glossopharyngeal nerve, which is the cranial nerve number 11. And the anterior tongue has a chorotympany nerve, which is a branch of cranial nerve number 7. So this is with human tongue. But my model organism, what I work in the lab, is a mouse tongue. So unlike human tongue, the mouse tongue has only one circummalar papilla at the posterior side of the tongue. So this one is a 3D image showing a circumvallate papilla of the mouse tongue, and each circumvallate papilla have this numerous taste bud. All this red structure, what you see, are numerous taste buds containing millions of taste receptor cells. So if I go deeper into the taste bud, as I said previously, taste bud has different taste receptor cells like type 1, type 2, type 3. Each of these can be characterized by the different markers they express, like type 1 express the antipydase marker or GLAST, type 2 express TRPM5 or G-alpha-gastasin, type 3 like STEP25. And below this, the taste receptor cells, there's also a type 4 cell, which we also call as the precursor cells. So precursor cells are also known as immature taste receptor cells, which in future will generate new type 1, type 2, or type 3 taste receptor cells. And outside the taste bud here, you can see there's the progenitor cells. So these progenitor cells are the ones that help in refueling of the taste receptor cells once the old one dies and the new one comes in. So previous study have shown that unlike all the other epithelial tissue, the circumvallate papilla has its own set of niche of LGR5 expressing stem cells here at the bottom of the trench. So previous studies have shown that during homeostatic condition, these LGR5 expressing stem cells give rise to all the taste as well as non-taste cells during homeostasis. So the trajectory, what it follows is that the LGR5 stem cell is present here, which can self-renew. These further differentiate into progenitor cells, which express the various markers such as K14, SOX2, and PCNA. These progenitor cells form the precursor cell, which express the sonic hedgehog. And finally, this precursor cell give rise to type 1, type 2, or type 3 taste receptor cells, which collectively express the marker keratin 8. So the turnover rate of each type of taste receptor cells was found out to be 10 to 14 days, after which the old cell dies and the cycle of renewal continues. However, the important factor here is this entire renewal of taste bud and taste receptor cells is dependent on the nerve. So this is a section view of a mouse circumvallate papilla. You can see here the red ones are the taste buds, and these are densely innervated by the nerve fibers. So because this is a circumvallate papilla, these nerve fibers belong to the glossopharyngeal nerve coming from the cranial nerve number 9. So previous study have shown that nerve has a very fundamental role in renewal of taste receptor cells. So if you look at the left, so this is a sham mice. So you can see there's multiple taste buds here on the trench of the circumvallate papilla. But if you cut the nerve which innervates the taste bud, the glossopharyngeal nerve specifically for circumvallate papilla, all the taste buds start degenerating by 14 days. So like any other epithelial tissue, there are two ways by which the damaged tissue can regenerate itself. One is by the use of adult stem cells, example LGR5, or like in the liver, just by proliferating of the remaining sister cells. But recent studies have shown that apart from adult stem cells or the remaining cells, there's another method by which tissues can regenerate. It is called as adaptive cellular plasticity. So one way is by transdifferentiation, and the second is by dedifferentiation, in which there's a reprogramming of differentiated cells during the regeneration phase. So for today's talk, I will mainly focus on dedifferentiation mode of regeneration of the epithelial tissue. So during normal homeostasis, you see there's a stem cell, it forms the progenitor cells, and these progenitor cells gives rise to the mature cells. That is, it follows a proper lineage hierarchy. However, after injury, during dedifferentiation, some of the mature cells or the progenitor cells can reprogram and revert itself back to stem cell. On the other hand, in transdifferentiation method, like one mature cell can give rise to another mature cell belonging to a totally different lineage. So these are the different methods adopted by the cells during regeneration of a tissue. So some of the example of dedifferentiation and transdifferentiation has been shown previously. So in tracheal epithelium, you see here, these are the basal stem cells which replenish the secretory cells of the tracheal epithelium. However, it was shown that after ablation of the basal stem cells, some of the secretory cells, which are the mature cells, can de-differentiate and give rise to the new stem cells during regeneration phase. Similarly, in the liver, after ablation of the biliary epithelial cells here, so if you ablate the biliary epithelial cells, the hepatocytes can transdifferentiate and give rise to new biliary epithelial cells during regeneration phase. So this is what we know till now during the different methods of regeneration. So coming back to my model system, the tongue and the taste bud. So we saw that after nerve transaction previously, the taste buds start degenerating. So this is a surgical view of the thoracic region of the mouse. So you can see here, this is the glossopharyngeal nerve, hypoglossal nerve, and the carotid artery. So to incur the injury to the taste buds and the taste receptor cells, I transected this glossopharyngeal nerve, which innervates the taste bud. So after transection, what I observed was, so this one is a sham mice in which the nerve was not cut. So you can see the taste bud structure is maintained and it's heavily innervated by the nerve fibers. However, after 2 weeks, the taste bud started degenerating. But surprisingly, what we observed was not all taste receptor cells were degenerated, but some of the taste receptor cells were still maintained in the circumvallate papilla epithelium. And these taste buds were maintaining in the absence of nerve contact. So there was no nerve contact happening with them, but still they were surviving. Furthermore, by 4 weeks following the nerve reinnervation, the taste bud started regenerating, and by 6 weeks, the taste receptor cells and the nerve density increased. So based on this data, the preliminary data, my question for the study was, what is the fate of these remaining taste receptor cells after injury? Do they have any role in regeneration of the taste bud? If they do, how do they regenerate? Do they undergo dedifferentiation into LGR5 stem cells? Or do they dedifferentiate into progenitor cells? Or they just self-duplicate and give rise to new taste receptor cells within the taste bud? Or they have no role in regeneration? So this is what my question was. So to answer this question, I went to the gold standard of identifying label-retaining cells, which is a BRD incorporation method, to identify if there's any population, special population of taste receptor cell within the taste bud. Now, BRDU, as you can see in the figure, is just an analog of thymidine. So instead of methyl group, it has a bromine group. And then because it resembles this thymidine nucleotide, it gets incorporated into the DNA of the cell during the DNA replication. So one of the important aspects of BRDU is that you can identify once the DNA is incorporated into the cell, the high proliferating cells will dilute the dye over a period of time, whereas the quiescent cell or slow cycling cell can retain the dye and can be observed over a longer period of time, having the BRDU dye in the DNA. So these cells which can retain the dye over a longer period of time are called as label retaining cells. So why label retaining cells are important is that in previous studies in the development of feather, it was found out that based on the orientation of the label retaining cells, whether it have a radial symmetry or whether it has a bilateral symmetry, it was easy to depict whether the feather formed will be a downy feather or whether it will form a flat feather. So this was for development. For regeneration also, when we do not know a proper stem cell marker, the label retaining method was able to identify, for example, here in the liver, some of the label retaining cells which had a role in regeneration, which activated after injury and expressed the marker for like stem cells or progenitor cells and participated in the regeneration of the liver. Hence, label retaining cells have a bit of importance in identifying a special population or a stem cell population whose markers are unknown to us. So using the similar approach, we injected the BRDU into the mice for 6 days at the interval of 12 hours apart and harvested the tongue at 6 weeks. So considering the normal turnover rate of taste receptor cells to be 10 to 14 days, at 6 weeks after harvest, we were able to see some of the type 2 and type 3 taste receptor cells being maintained in the taste bud, so which was much greater than the expected lifespan. So indicating that there is a population of long-lived taste receptor cells in the taste bud. So the next question was whether these long-term liver-retaining cells have a role in regeneration. So similarly to the previous experiment, we injected the BRD into the mice. We waited for 4 weeks, performed the nerve transaction, and harvested the tongue 6 weeks after the nerve transaction. So even during this time, we could observe that some of the type 2 and some of the type 3 taste receptor cells were still maintained in the taste bud, indicating that these liver-retaining cells did not degenerate after the nerve injury and were maintained independent of the nerve contact. Furthermore, by double labeling experiment, that is by using BRDU and IDU, which is also a DNA incorporating dye, we observed that these BRD positive label retaining cells during regeneration incorporated IDU, indicating that the post-mitotic taste receptor cells, which are the functional differentiated cells, upon injury re-enter the cell cycle. So this gave us our first clue that maybe the differentiated taste receptor cells might have a role in regeneration of the taste bud. So previous study had shown that these LGR5 expressing cells, which are present at the trench of the circumvallate papilla, gave rise to all the taste cells like type 1, type 2, type 3, as well as non-taste cells during injury, during the regeneration phase. So we employed the similar Cree-Loxp system to identify whether all the cells indeed come from LGR5 expressing stem cells or no. So we used the mice LGR5 GFP, and by injecting the tamoxifen, the CRE was activated, which led to excision of the stop codon and expression of the TD tomato signal. So based on the previous result, this is what is to be expected, that LGR5 stem cell will be red color, the progenitor cell will be red color, and all the cells which come from the LGR5 will be red color. So this is what the previous result said. Interestingly, what we observed was after the GLX, after the nerve transaction, during the regeneration phase, the regenerated taste bud, not all the taste buds were derived from the LGR5 expressing cells because they did not express the TD tomato signal, both at the 6 weeks as well as 4 weeks. So instead of getting a full red color taste bud, we observed this mosaic expressing a mosaic color taste bud in which some cells were red color, some cells were not. So this data indicated to us that not all cells during regeneration come from LGR5 stem cells, but there is another pool of cells which contribute to regeneration of the taste bud. So our next question was, if not the stem cell, maybe the progenitor cells participate in regeneration of the taste bud. So following the similar Cree-Loxp system, we lineage traced the K14 progenitors progeny, and we found out that in this case also, not all taste receptor cells during regeneration at 4 weeks and 6 weeks were coming from progenitor cells. So indicating that apart from LGR5 cells and K14 cells, still there was another pool of cells where that was helping in regeneration of the taste bud. So this was another study published in 2013 for the tracheal epithelium. So in this study, they showed that there's a stem cell population here expressing cytokeratin 5, the red color here, and then there's differentiated cells here which express the YFP, the green color. So in the sham mice, there's a clear separation between these two cell population. But after injury, what they observed was some of the differentiated cells were going back and giving rise to the injured stem cells. So as you can see here by the co-localization staining here. So we asked the next question, whether this kind of similar mechanism happen in taste bud also. We did the staining, we saw that. So this is a sham mice. So you can see sham mice has a keratin 18 expressing differentiated cells and keratin 14 progenitor cells which surround the taste bud. So there's a clear separation between the differentiated cell as well as the progenitor cells. Interestingly, after injury, we observed that some of the surviving cells at 2 weeks, they were expressing marker for both the differentiated cell as well as the progenitor cells. This phenomenon was much evident at 4 weeks, as you can see that some of the subset of taste receptor cells were positive for both the keratin 8 as well as keratin 14. This gave us our idea or the result that differentiated cells, which express the K8, they acquire progenitor cell-like properties, which is the K14 positive, after the nerve injury. So the next question was whether this K8 positive, K14 positive taste receptor cell can regenerate the taste bud. So to identify that, we again went back to our Cree-Roxby system lineage tracing. So in this case, the Cree was driven by the promoter under K8, such that after injection of tamoxifen, the stop codon will be excised out, and all the remaining taste receptor cells will be labeled red. So by this, we can see if there's any regeneration happening because of the surviving taste receptor cell or no. Interestingly, what we saw was that after GLX, some of the K8 or the differentiated cell, K8-derived cells, which are expressing tree tomato, were proliferative. Like here, they expressed the marker PCNA. Furthermore, they were basally located, which expressed the marker K14, and also they were expressing marker for SOX2, which is a marker for progenitor cells of the circumvallate papilla. So with this, we said that the K8-derived cells were proliferative and expressed progenitor-like potential and might have a role in regeneration of the taste bud. Furthermore, we checked for the regeneration of the taste bud at different time points. So if you look at the sham mice, the K8-derived taste receptor cells are located only inside the taste bud. However, during regeneration, the K8-derived cells, tree tomato cells, not only regenerate the taste bud, but also some of the non-taste epithelial cells here and like the progenitor cells here, indicating that after injury, the differentiated cells had acquired some kind of plasticity and hence is able to differentiate or regenerate the taste bud as well as non-taste epithelial cells during regeneration. So to identify this progenitor or stem cell-like potential of these K8 differentiated cells, we next perform the organoid formation assay. So now organoids can be formed only by the stem cell or the progenitor cells, and differentiated cells cannot form organoid. So we performed the GLX and injected the tamoxifen to label the surviving taste receptor cells like this. We harvested these red color cells, surviving taste buds, and put it into the matrigel. And indeed, these surviving taste receptor cells did form organoid once cultured in vitro. Furthermore, these organoids contain the basal cells, the progenitor cells, K14, SOX2, as well as they were proliferative. Not only that, these organoids also contain the differentiated cells, indicating that this may be a functional organoid which contains both the progenitor cells as well as the differentiated cells. So till now, we know that the taste receptor cells were undergoing dedifferentiation, but it was not known whether this kind of dedifferentiation is a stable, which means it's going back to a stem cell state, or is it just a transient dedifferentiation, which is going to a progenitor cell state. So in this paper published in 2022, they said that to identify the different kinds of dedifferentiation, chase the lineage for a longer period of time. If a cell is undergoing a transient dedifferentiation, like here for keratin 20, over a longer period of time, the tree tomato signal is lost because progenitor cells have a shorter lifespan compared to stem cells. And if a cell is undergoing dedifferentiation into stem cell, the progeny can be observed over a longer period of time because stem cells renew the cells throughout the life. So following this similar reference, we performed the experiment. We injected tamoxifen, performed the nerve transaction, and harvested the tongue at 13 weeks. So indeed, we saw that after 13 weeks, no TD tomato positive cells were observed in the regenerated taste bud, indicating that the K8 or the differentiated taste receptor cells were just transiently de-differentiating and giving rise to new taste receptor cells. And after the taste receptor cells are generated fully, they just disappear or they degenerate. So in conclusion, we saw that after injury, some of the differentiated taste receptor cells, they de-differentiate into progenitor cells, give rise to regenerated taste bud, and then they disappear. So in summary, I would like to say that although during the homeostatic condition when nerves are connected, the LGR5 stem cells are the main players in giving rise to new taste receptor cells during homeostasis. But during regeneration, along with LGR5 stem cells, some of the differentiated taste receptor cells can also undergo de-differentiation and give rise to regenerated taste bud. So I would like to thank my professor, Professor Han Sung Jung, for this wonderful opportunity, and all my lab mates for their constant support. Thank you everyone for listening. I'll be happy to answer your questions.